Welcome to biotechnology.com. In this tutorial we are going to discuss about the different plotting techniques, definition and also some major important points. According to central dogma of biology, a gene is a segment of DNA that codes for a functional polypeptide. In a molecular biology lab, we may be looking at a specific DNA. For expression studies, we may be looking at an mRNA or a polypeptide. The technique to find out a specific DNA in a sample is called as a southern blotting, whereas for expression studies we may look either at mRNA or a protein. The technique to find out specific mRNA is called as a northern blotting, and also in the case of a protein, the technique to find out a specific protein in a sample is called as a western blotting. So we'll be looking into some aspects of southern blotting. The technique was developed by E.M. Southern in 1975. It's a technique used to detect the presence of a particular DNA fragment in a sample. The principle of this technique is hybridization. We are using a radio-labeled or any labeled probe in order to find out the target DNA. This probe can bind to the specific DNA target and form hydrogen bond so that we could easily recognize as it is labeled. The two important aspects of hybridization is that the reaction is highly specific. The probe will only bind to the complementary sequence. Second thing, even if there are millions of molecules, DNA molecules right there, the probe is capable of finding out our target DNA. Northern blotting is a technique for detecting specific mRNAs separated by electrophoresis by hybridization to a labeled DNA probe. Here also, the technique was developed by Alvin et al. In 1979. The principle is the same as southern blotting. Hybridization is the key, where we will be using either a DNA or RNA probe. In the case of western blotting, it's a technique to find out a specific protein. Usually, the electrophoresis is STS page, as protein must be denatured and it must be uniformly charged with STS. Principle, it was developed by Tobin in 1979. The principle is antigen-antibody interaction and is highly specific. Our protein can be considered as an antigen, and we will be adding a primary antibody that is followed by a labeled antibody, so that this antibody will bind to the specific antigen. If our protein of interest is present in the sample, we'll be getting some signals. There is one more alternative for southern blotting, which is called as dot plot technique. Here the electrophoresis step is not there. Samples that directly applied onto the membrane, nitrocellulose membrane as spores. The advantage of this technique is that we can run a number of samples in a single test run, and the procedure is simple compared to southern blotting. Principle is hybridization. Here also we are adding a probe, and we'll be washing out unbound probes, and probes are radio-labeled. If the DNA is present, then the probe will bind to the DNA and we'll be getting some signals. Hope things are clear, and you are with biotechnology.com. Thank you so much for watching.